Good morning, one and all. I'm Dr.S.Selvakumar, Department of Agricultural Biotechnology, Bharat Institute of Higher Education and Research. So I welcome you all for my plant molecular biology class. So subject code of the subject U18Pcac401. So subject name plant molecular biology. What is the topic of today's class? Gene regulations in the prokaryotes and eukaryotes. Now we're going to the class. First, what is gene expression? So the gene expression is the process by which the information encoded in a gene is used to direct the assembly of a protein molecule. So the gene expression is explored through a study of the protein structure and protein function, transcription and transcription. So the differentiation and the stem cells. So it is a process by which information from a gene is used in the synthesis of a functional gene product. Is called gene expression. So the gene expression continues. These products are often proteins, but in non-protein coding genes such as ribosomal RNA, that is rRNA, transfer RNA, that is tRNA, or small nuclear RNA, that is snRNA genes. The product is a functional. Right. The process of gene expression is used by all known live eukaryotes, including the multicellular organisms. In case of prokaryotes, bacteria and archaea. Next, I'm coming to what is the regulations of gene expression. So the regulation of gene expression includes a wide range of mechanisms that are used by the cells to increase or decrease the production of specific gene products, that is proteins or RNA, ribonucleic. And next part, the gene regulation is essential for viruses, prokaryotes, and eukaryotes. As it increases the versatility and the adaptability of an organism by allowing the cell to express the protein when needed. Although a functional and gene product may be an RNA or a protein, the majority of known mechanisms regulate the protein coding genes. Any step of the gene expression may be modulated from DNA to RNA by transcription to the post-transcriptional modification of the protein. The first discovered example of a gene regulation system was the lactoferrin, that is lactoferrin. So this is discovered by Jacques Monod, in which the protein involved in lactose metabolism are expressed by Escherichia coli, that is E. coli, only in the presence of lactose and the absence of glucose. Next, I'm coming to the regulations of the gene expression continues. So the gene regulation derives the process of cellular differentiation and morphogenesis, leading to the creation of a different cell types in multicellular organisms, where the different types of cells may possess the different gene expression profile. Example 2. What is genes? The genes are the subunits of DNA. What is genes? Genes are the subunits of DNA. So, the information database of a cell that is contained inside the cell nucleus is called. So, this DNA carries the genetic blueprint that is used to make all the proteins in the cell needs or the cellular. So every gene contains a particular set of instructions that code for a specific protein. Underline, every gene contains a particular set of instructions that code for a specific protein. Next, I'm coming to the classification of genes based on the expressions. Number 1, the constitutive genes is also called housekeeping genes. Housekeeping. These type of genes are expressed at a fixed rate and irrespective to the cell conditions. The second important point, the structure is very simple. If I'm coming to second class, the controllable genes. What is controllable genes? The controllable genes are expressed only as needed. Their amount may be increased, sometimes decreased, with respect to the basal level in the different conditions. The controllable gene structure is relatively complicated with some response elements. Underline some response elements. Next, so the types of gene regulations. There are two different types of gene regulations. One is called positive regulations. The second one is called negative regulations. What is positive gene regulation? The case of positive gene regulation, when the expression of genetic information is quantitatively increased by the presence of specific regulatory element, is known as positive regulation. The element modulating positive regulation is known as activator. Underline, activator or positive regulator. Do not question what is activator of the gene regulation, or what is the activator of the gene regulation, or what is the positive regulator of the gene regulation. The element modulating positive regulation is called activator or positive regulator. Next, I'm coming to the negative regulation. When the expression of genetic information is diminished by the presence of specific regulatory element, is known as negative regulation. In the case of negative regulation, the element or a molecule mediating the negative regulation is called repressor. So activator, repressor. Next, I'm coming to what are the steps which are basically involved in the gene expression. One by one we'll see. Number one. The synthesis of primary RNA transcript. Synthesis of the primary RNA transcript. That is second one. Post-transcriptional modifications of mRNA. What is mRNA? Messenger. So the post-transcriptional modifications of mRNA. 3. The messenger RNA degradation. Messenger RNA degradation. 4. The protein synthesis. Protein synthesis, that is translation. Next one. So, post-translational modifications of proteins. Post-translational modifications of proteins. Next one coming to the protein targeting and transport. Protein targeting and transport. Next one coming to the protein degradation. Protein degradation. Next one coming to the prokaryotic gene regulations. Prokaryotic gene regulation. In prokaryotes, the primary control point is the process of transcription initiation. The prokaryotes, the primary control point is the process of transcription initiation. There are different ways for the regulations of the gene expression in bacteria. The regulation of gene expression can be done by some operon pathways, such as number 1, lag operon, number 2, tryptophan operon, number 3, transcriptional control, translational control, and post-translational control. Next, I'm coming to the prokaryotic gene expression regulation steps, the important steps. So, transcriptional control, translational control, post-translational. Next, I'm coming to very important operons. What is operon? Basically, in genetics, a operon is a functioning unit of, underline, functioning unit of genomic DNA containing a cluster of genes under the control of a single promoter. So the operons occur primarily in the prokaryotes, but also in some eukaryotes. Operons are related to the regulations, stimulants, regulants, stimulants, and modulants. The operon basically made up of several structural genes arranged under a common promoter and regulated by the common operator. So common promoter, common operator. So it is defined as a set of adjacent structural genes plus the adjacent regulatory signals that affect the transcription of the structural genes. Next, I'm coming to operon structure. Coming to operon. And operon is made up of 4 basic DNA components. There are 4 basic DNA components. What are the 4 basic DNA components? Number 1, the promoter. 2 more questions. What is promoter? A nucleotide sequence that enables a gene to be transcribed. Underline, a nucleotide sequence that enables a gene to be transcribed. Then, the promoter is recognized by the RNA polymerase. What is RNA polymerase? The group of RNA which the initiates, which then initiates the transcription. So promoter is nothing but a nucleotide sequence that enables the gene to be transcribed, and the promoter is recognized by the RNA polymerase, which then initiates the transcription. The first. The second basic DNA component are regulator. What is a regulator? These are the genes. Regulators are the genes. So these genes control the operator gene in cooperation with certain compounds called inducers. And co-repressors are also present in the cytoplasm. So the regulator may be defined as the genes control the operator gene in cooperation with certain compounds called inducers and co-repressors present in the cytoplasm. Second one, the structure of operator. Third basic component of the operator. What is operator? A segment of DNA that a repressor binds to. A segment of DNA that a repressor binds to. So it is classically defined in the Lake operon model system. So the Lake operon has the segment between the promoter and the genes of the operon. The genes of the operon. So the operator genes may be defined as a segment of DNA that a repressor binds to, and it is classically defined the Lake operon as the segment between the promoter and the genes of the operon. So, the fourth basic molecule of the structural genes. The genes that are co-regulated by the operon. Two structural genes. The genes that are co-regulated by the operon. The regulations of the operon. The operon regulation can be either negative or positive by the induction of repression. The induction or the repression. The negative control involves the binding of a repressor, underline binding of a repressor to the operator to prevent the transmission. So, in the negative inducible operons, it's a regulatory repressor protein, is normally bound to the operator, which prevents the transcription of the genes on the operon. In continuation of operon regulations. If an inducer molecule is present, it binds to the repressor and changes its conformation so that it is unable to bind to the operator. This allows for the expression of the operon. The late operon is negatively controlled inducible operon, where the inducer molecule is allolactose. Underline, the late operon is a negatively controlled inducible operon, where the inducer molecule is allolactose. Next, so continuation of operon regulations. In the negative repressible operon, the transcription of the operon normally takes place. Negative repressible operons, the transcription of the operon normally takes place. The tryptophan operon involved in the synthesis of tryptophan, note, which itself acts as the co-repressor, and it is a negatively controlled repressible operon. So TRP operon is called negatively controlled repressible operon. Next, I'm coming to in continuation of regulations of operon with positive control. With the positive control, and activator protein stimulates transcription by binding to the DNA. In the positive inducible operons, the activator proteins are normally unable to bind the pertinent DNA when an inducer is bound by the activator protein. It undergoes a change in the conformation so that it can be bind to the DNA and activate the transcription. Next, I'm coming to in continuation of the left operon. In the positive repressible operons, the activated proteins are normally bound to the pertinent DNA segment. However, when an inhibitor is bound by the activator, it is prevented from the binding to the DNA. This stops activation and transcription of the system. Underline. However, when an inhibitor is bound by the activator, it is prevented from the binding the DNA. This particular process stops the activation and transcription of the system. Next, I'm coming to lactoferron, that is lactose operon. The lactoferron of the model bacterium E. coli was the first operon model system to be discovered and provides a typical example of operon function. It consists of 3 adjacent structural genes, that is a promoter, a terminator, and is called an operator. So the 3 adjacent structural genes are number 1 the promoter, terminator, and number 3 operator. The leg operon is regulated by several factors, including the availability of glucose and lactose. Next, I'm coming to TRP, tryptophan, TRP. It is discovered in 1953 by Jacques Monod and his colleagues. The TRP operon in E. coli was the first repressible operon to be discovered. Next, this particular TRP operon contains 5 structural genes: TRP-E, TRP-D, C, and TRP-B and TRP-A. So 5 structural genes, which encodes the tryptophan synthetase enzyme. The operon also contains a promoter which binds to RNA particles and an operator which blocks the transcription when bound to the protein synthesized by the repressor genes, that is TRPR. The TRP repressor protein gene, TRPR, that binds to the operator. Next, I'm coming to eukaryotic gene regulations. So the eukaryotic cells have much larger genome. The eukaryotes have much greater cell specifications. Therefore, the eukaryotic cells contain an enormous amount of DNA that does not program the synthesis of RNA or protein. This eukaryotic gene regulation requires a complex organization. Next, in the case of eukaryotes, the expression of gene into proteins, it can be controlled at various locations. Next, the eukaryotic gene expression checkpoints. The synthesis of proteins is controlled right from the chromatin stage. So the expression of the gene is controlled at many steps during the process of transcription and translation. The description of the control points is built in detail the subsequent steps. So, the main steps involved in a eukaryotic gene expression. I tell you one by one. The transcriptional control, number 1. Number 2, the RNA processing control. RNA processing control. Number 3, RNA transport and localization control. 4. The translational control. Translation control. Number 4. mRNA, that is messenger RNA, degradation control. Next, 6th one, the protein activator control. Next, the structure of the. There are two forms of chromatin. One is called euchromatin, second is called heterochromatin. What is euchromatin? So euchromatin is a simple coil, very lesser, transcriptionally active regime. So very simple coil, transcriptionally active regime, which can be easily accessed by the enzyme RNA polymerase. Whereas the second one is called heterochromatin. A highly contains, tightly packed, transcriptionally inactive region. Euchromatin means transcriptionally active region. Simple coil. Heterochromatin highly contains, tightly packed, but transcriptionally inactive region. The genes in this region cannot be accessed by the RNA polymerases for the active transcription. Right. Next, coming to ubiquitination. H2A, H2B, H2A for transcriptional inactivation, H2B is the transcriptional. Next, I'm coming to the regulation of transcription. The basal promoter or core promoters, these promoters, base promoter or core promoters. So these promoters reside with the 40 base pairs upstream of the static site. So these promoters are seen in all protein coding genes. Underline, all protein coding. The second one is called upstream promoters. These promoters may lie up to 200 base pair upstream of the transcriptional initiation. The structure of this promoter and associated with binding factors and keeps varying from gene to gene. So, enhancers. What is enhancer? What is enhancer? The enhancers can be located upstream or downstream or within the gene that is transcribed. The binding of these enhancers with the enhancer binding proteins, that is called transcriptional factors. And it increases the rate of transcription of that gene to a greater extent. Next, I'm coming to in continuation of the enhancer. The promoters are capable of initiating the lower levels of transcription. Next, the enhancers are responsible for the cell or tissue specific transcription. The final one, each enhancer has its own transcription factor that it binds to.